first speaker, my friend Frédéric Reva, the CEO of Geneton. And as we have after many scientific presentation or technical presentation, I asked Frédéric to give a global overview on the segment, the industrial segment, scientific segment, because there are many challenges. Scientific challenges are very well managed by many scientists around the world, but after that, there are many considerations and conditions to have those discoveries transferred to the market. So Frédéric is a specialist because he's a scientist, but now he's managing Geneton, which became a large organization, including in manufacturing. So Frédéric, if you can start, you have 15 minutes to introduce the subject from the global view.
Thank you, Christian. I will take my mask off just for the time of the presentation. Thank you for inviting me, Christian, and for giving me the opportunity of giving some introductory remarks here. So my name is Frédéric Reva. I'm CEO of Geneton, and just to tell you where I'm speaking from, just let me give you some elements about Geneton in a nutshell. I mean, we have been starting at Geneton Gene Therapy in 1997, and I usually say that I'm one of the oldest guys in the room involved in gene therapy, representing at least one of the oldest organizations involved in gene therapy. We have some specificities. We are a not-for-profit R&D organization that was started by a patient organization, the French Muscular Dystrophy Association. We are an integrated R&D center. We go from translational research to clinical validation. We started gene therapy in 1997 after being involved in genomics and establishing the first maps of the human genome. And we have initiated biomanufacturing since 2006, and I'll come back to biomanufacturing, which is an important part and a very specific hurdle to the field that we are interested in today. We have today 9 products which are in clinical trial, which either stem from our internal R&D or from collaborations. We have 9 other programs that should reach the clinic in the coming 3 years. We're very proud of some very iconic products in the field of gene therapy. Let me talk about Zolgensma, which is a Novartis product, but in which are embedded some founding patents from Geneton. So Novartis operates under a license from Geneton for this program. The program Lumevoq, the product Lumevoq, which is on its path for registration in Europe by the company Gencyte, is a program that was initially developed by Geneton together with the Institut La Vision here in Paris, and for which we did not only preclinical development, but also initial manufacturing. The product by Audentes and Astellas, the AT132 for myotubular myopathy, was also a product that was initially designed at Geneton, and we're very proud of our industrial partnerships with Roche, Novartis, Orchard, Sarepta, etc. Now, speaking from that position, of course, and having been involved in the field for now almost more than 20 years, we do see with high satisfaction the field blooming and cell and gene therapies that are now a commercial reality, with several products having reached the market since 2015, with Strimvelis for immune deficiencies, with Zynteglo for beta thalassemia, with Zolgensma for spinal muscular atrophy, Lumevoq on its way for optic neuropathy, Luxturna, retinal disease also, and as well as several CAR-T programs, which, depending on where you stand from, you could consider either as ex vivo gene therapy or cell therapy. In addition to that, more than 30 products are today in phase 3, both for rare genetic diseases and cancer indications. And we see these products we see this blooming of products. And I think for both scientists and industrials involved in the field, it is a source of intense pleasure and satisfaction. I can tell you that when we started gene therapy in '97 at Genethon, and for more than 15 years then, there were very few organizations, some in France here, but very few organizations worldwide that were interested in the field, both from a scientific point of view, from a development point of view, or from a financial point of view. Everybody did consider that this field was really too risky to capture investment, that it wouldn't work except in very specific indications. And now we do see more and more products getting to the market, not only for rare indications, but starting also for cancer and more frequent indications. But now the question is, how do we build from there a wide access to cell and gene therapy products? And as Christian said, it is not only a question of excellent science. And we will hear during this roundtable some very thrilling presentations about really forefront research, new products, new ways at looking at gene therapy. But building a sustainable pipeline and a sustainable portfolio of gene therapy products is not only a question of science, it's really a question also of approach and a question of technology. Let me try to address what these different challenges are in order to provide wide access to a large number of patients to these products. First challenge I want to stress is the manufacturing challenge. The manufacturing challenge, let me illustrate it by the panel which is on the left of the slide here, is a question of dose for gene therapy. I could have illustrated also by the question of logistics for CAR T cells, but I really chose to illustrate it by the question of dose for viral gene therapy. So we often heard when we got involved in manufacturing that, after all, other industries had been involved in manufacturing viruses, and in particular the vaccine industry. But if you look at the dosing aspect, you will see that a dose of vaccine is approximately 10 to the 6 viral particle per dose. If you get now to gene therapy for retinal disorder, in which you, in fact, inject in a very localized area, you go up to 10 to the 11 viral particle per dose. Now, if you look at neuromuscular indications, for which there's a really promising developments going on, the number of particles you need is 10 to the 15 to 10 to the 16 viral particle per dose. So we're talking here about a 1 billion to 10 billion fold increase in the number of particles per dose. And of course, there's no way you do that scale up by just doing more of the same and using more of the same technologies that you use for vaccines. And meeting this dose challenge is really a critical element in not only allowing the development of new products, getting those products in the clinic, but then also in providing access to patients once the product is on the market. It is also today one of the key elements of the cost of these products, and you probably heard that, for instance, for Zolgensma has been tagged the most expensive drug on the market, which is the case. I mean, it's the most expensive treatment on the market. Now, addressing the dose issue can be done from different angles. It means science in the field of virology, design better vectors, improved expression cassettes, novel capsids, and with the objective of injecting less product, having to inject less product because you will increase the efficiency of those products. But also, it means improving the bioprocess productivity. And this is a science in itself. Working on bioprocess is really innovation. It means not only mastering first-in-class technologies, it means increasing yields, it means cost reductions, but it means also disruptive technologies. It's not doing more of the same, it's not only improving whatever exists, but it's also disruptive technologies, trying to, let me be a bit technical, trying to get out of the traditional HEK293 cells to produce these viruses, find other ways, new types of cell-less elements, try to improve the purification methods also. I will not go into the technicalities, but clearly here, innovation is important. Now, in order to get really build upon the momentum that was given by the first products getting to the market, we need to think about next generation products and really about precision medicine. This means enhanced efficacy of the products. As I said, this is on the left panel. It means improved bioproduction, and the objective really should be an increase in 100 in the productivity of these methods for production. It means also being able to redose the products. You know that these gene therapy products are a one-off therapy with very spectacular results, but clearly some of the patients cannot be treated, are not eligible because they have existing immune response for the vectors, and also once you dosed, you cannot redose because you do stimulate the immune system. And clearly here, working on the immune response, developing strategies to bypass the immune response will allow to redose the products. And by redosing the products, you will come with therapies that are not only one-off, but can be repeated treatments, and at one point that may become chronic treatments. And from the pharmacoeconomics standpoint, you will have products that will look much more like traditional products from a pharmacoeconomic standpoint, rather than these products for which the companies try to make a profit just on one single injection. And this takes me, as a conclusion, to my last point. If we want to build a sustainable industry in cell and gene therapy, we have to think about the pricing strategies. We have to think of whether cell and gene therapy can sustainably remain the most expensive drug treatments around. This is the case today, and there might be a certain acceptance by the payers. Systems are not the same in the United States and in Europe. There could be a certain acceptance for the first products, but my strong belief is that on the long run, the systems will not be able to support the cost of such high costs and such expensive drugs. We have to rethink the pricing strategies. A 2 million injection is something that might work in the short while, will not work on long term, and working on cost of goods, work on re-injection is a way of altering the economics equation, but clearly this is a factor that as an industry we do have to address. Having said this, I thank you, Christian, and I leave the floor to the next speaker.
So I think to start with those questions is a good start, because it will be certainly something you will develop during your scientific speech. If you forget what has been said, perhaps we'll have problems to have drugs, in the market. So it's good. You need to manufacture and to price it, Okay, so we start with the first first part of this session, so concerning gene therapy. And I shall ask to Nathalie Cartier.
So before we get started with the slides, maybe I can introduce myself. So first, thank you very much for the invitation to give this talk this morning. So the question is quite complicated to address. Where do we stand in gene therapy now? So I will try to make some comments about around this question on my domain, which is neurological disease. I'm MD. I'm heading a research lab at the Paris Brain Institute in Pitié-Salpêtrière Hospital. And I've been doing gene therapy for 20 years, from basic research to clinical trials. And I have tried to accelerate these development steps towards clinical trials by starting a company called Brain Vectis. And through that, we are going to start a phase 1 clinical trial for a very severe disease called Huntington disease next year. So this is to show you how you can manage this research in gene therapy from the very early stage to clinical applications, and how it moved from the last, let's say, 20 years, from the first clinical trials to where we are now. And thanks to what said Frédéric, I will probably, again, repeat some of these things about manufacturing, about next generation strategies and everything. But I think it's really the important points today to show you that, sorry, indeed, gene therapy is real medicine today, and it's going to be much more in the future years. So I think there is a problem with my computer. Is it okay? Ah, okay. Can you go to the first slide, please? Can you go? Est-ce que vous pouvez aller à la première diapositive, s'il vous plaît? Okay. So this is the title. This is what I just explained. So should I take that, or no? You do it. So next slide. So next slide. Next slide, s'il vous plaît. Okay, so this is the long road that we took from the beginning, the very beginning of gene therapy, when and the first step, when at least at the beginning DNA structure was identified, and first use manipulation of DNA allowed to make the very first step of gene transfer. And this shows you the first step from, I don't know. Okay, the very first step from, as you see here, the first trial with SCIDX patients, then the retinal. Can you go back? I've not done anything. Okay, the retinopathy treatment and the first clinical trials using HIV vectors that we've been able to do. I will say a word about that. And then the first successes around the 20. Could you please go back? Okay, the first success with thalassemia or hemophilia, and more recently, all the work about CAR T cells and very successful treatment for cancer research. So, next slide. Okay, so what are the tools to do so? There's nothing better than viral vectors, as you know. Move forward, next, click. Okay, so this, and click. So these viral vectors are able to bring genes, therapeutic genes, into the cells, and this can be either in an episomal stage, so non-integrated, or integrated with lentiviral vector. And this is what it carries. It carries DNA or RNA. Next slide. So there are two things that can be done. One is bringing directly the viral vector, carrying the therapeutic gene into the body, which is in vivo gene therapy. And the other approach is called ex vivo gene therapy, where you take cells from the patients, and you put in contact a viral vector with these cells to correct them and so they express a therapeutic gene, and then you put the cells back to the patients. Next slide. Next slide. Okay, so if I go now to what I know about, which is central nervous system, brain gene therapy, there's two ways to do this, exactly what I said. Click and click. So either ex vivo gene therapy, which is to take the cells, and particularly hematopoietic stem cells, that you can manipulate, in which you can put a therapeutic gene and put the cells back to the patient, and these cells will be able to migrate to the central nervous system, to the brain, and locally to differentiate into cells that are macrophage-derived, which are called macroglia, and that will bring a therapeutic protein and correct several diseases. So this is the kind of ex vivo gene therapy that is done for CNS. And the other way, of course, is taking the gene directly into the body, and particularly by direct injection, as you see here on the right part in the head, bringing directly the gene by local injection into the brain or delivery into the spinal fluid. Next slide. So the first example I want to show you of how we have done that was the first use of an HIV-derived vector to treat a very severe disease of the children called adrenoleukodystrophy. And this is, oh no. Could you go back, please? Okay, this adrenoleukodystrophy, and this is a work we've been doing with Alain Fischer and Marina Cavazzana. And we've been able to use this HIV vector completely free of any toxicity, of course, to bring back the therapeutic gene into the patient and giving back the autologous transplanted cells in this patient to cure this disease. And this is what you see on the right part of the slide, where you see two children with, one children on the right with very severe lesions and two treated patients with completely stabilization of the disease. So this was really a very strong improvement in this gene therapy strategy. Next slide. This was developed by Bluebird Bio, and the phase 3 study was completed recently and published a few years ago, and this allowed very few days recently to announce that they have got the marketing authorization in Europe. So really this is a major step, of course, for gene therapy for these kind of very severe diseases. Next slide. So the other strategy relies on other type of vector called AAV vectors. And AAV vectors are what you see here. They are DNA vectors. They are not integrative vector, and they really are kind of toolbox because there are many serotypes, and this is what is summarized on the right part of the slide. Many serotypes allowing different specificity for different tissues, different type of cells, and this can be modified, this can be improved, this can be manipulated to improve the specificity, improve the efficacy, and this is today really a major tool for gene therapy and for the future. Next slide. And if I go back to, with this vector and to the field of the brain disease, one of the first trial, one of the first application for very severe disease has been what is shown here for one severe lysosomal disease. And this showed that bringing the gene, as I said before, by direct injection, so a quite invasive strategy, of course, direct injection into the brain, could stabilize this patient affected with very severe lysosomal disease. And on the next slide, you will see that this was published quite a long time ago now. It was published in 2012, but it showed that after 10 years, safety demonstration could be demonstrated, which again was a major step in this kind of strategy, very aggressive. And also because you know that gene therapy is one time, the gene is there, you cannot take it out, and you really need to show safety because it's present for a lifetime. So this was a major step too also because it showed that it was safe and it could bring some efficacy with long-term expression. Next slide. So I don't go into details of the many rare diseases for which the same strategy has been used with some results, but I go to Parkinson's disease because it's, of course, a common disease and an important step also for gene therapy. There are several trials ongoing for Parkinson's disease. Parkinson's disease affects this region of the brain that you can see on the right part in blue. And if I show that, it's because you will see on the next slide an example extremely striking. This is the equivalent, we may say, of Parkinson's disease as a genetic disease, is a deficient of the enzyme called AADC. And it affects exactly the same regions as I've shown for Parkinson on the last slide. It's a genetic disease. It's very severe. And you will see on the next slide, please, that children affected with this disease, next slide, please, are extremely affected. They don't speak, they don't eat alone, they don't stand, and they make no progress. A click, please. So you see this little girl with absolutely no contact and no capacity to be autonomous. Next slide. After gene therapy, one injection, one single injection, this is the same little girl 1 year after. Click, please. And you see that now she's able to move, she's able to speak, she's able to eat. So major progress also. Next slide. So this is based again on direct administration. Of course, this is complex. This is not feasible for every disease, and this is not feasible for every patient to treat hundreds of patients. So one new step after that has been important one, has been the use of IV delivery, intravenous delivery. One single injection again, but intravenous delivery. And this was for another very severe disease that you see the child on the right part up there, called spinomuscular atrophy. Extremely dramatic disease. Children don't sit, don't walk, of course, cannot eat alone, and die in the first years of life. And next slide. What has been done by Jerry Mandel a few years ago was to do one IV, one intravenous injection of the gene into these little children. And this showed that, this was published in the New England Journal of Medicine, and the curve that you see here is the improvement of all children, so the development, normal development of all children. The only, down on the slide on the right is a child treated late. Again, meaning that you need, of course, to treat early this very severe disease. But next slide, what it showed is that with only one administration, these children can be saved, and these children can develop normally. So again, a second major improvement for gene therapy. Next slide. And this was approved, of course, and this treatment is now on the market to treat these children. Next slide. So, few examples showing that it's very encouraging. It's complex, we are not at the end of it, but it's very encouraging. So where are we now? What do we need? I took three things, because we cannot go into everything, but I took three things that I feel important. One is IV delivery. I just said about that. I think it's important to have simple way of administration of any therapeutic product. For brain, what are the next steps? I'll say just a word on that. And I want to stress on something which is complex disease, because what I've talked about are genetic disease, rare disease. It's simple. It's simple theoretically. One gene is missing, you put back the gene, and you hope to treat the patient. But it's not the only medicine, of course. And gene therapy is not only for rare disease. Gene therapy can treat every disease based on the comprehension of the pathology of the disease and the pathway that you have to target to treat the disease. And this is what I put on the right part. So one of the major questions we have today is understand the disease pathways to be able to develop new targets for gene therapy. So what we need? Toolbox, and Frédéric has said a lot on that, and manufacturing. Manufacturing is a critical issue, and there's a lot done these days and a lot of progress. Toolbox, I'll say just a word, and understand the pathology. Next slide, please. So toolbox. The vectors. I showed you AAV as a really good tool. Next slide. And these tools can be extremely optimized. You can modify everything. You can turn it in every way and have it very specific. Hopefully having it able to go IV and to cross the blood-brain barrier, for example, to go to the CNS, and also to be very specific to tissue. Next slide. And also you can work on the gene you put in the vector. So it's a cassette. In the cassette you have the gene, but you have all the sequences that allow to go in specifically into one cell, or not to go in other cells, and these kind of things. And it's a kind of, next slide, of mixing where you put all these sequences together, and then you select the best, and then you go to the clinic with it. So it's really what is done all over the world in different teams to go to clinical application. Next slide. Toolbox 2 is manufacturing. So you've heard about that just before, and manufacturing is key. Why? Next slide. Because it's okay to do one injection of few microliters of a product like here for the retina, Luxturna, in the eye. But how do you do to treat disease where you need to cover all the body, like all the muscles? Next slide. And the example is the Duchenne myopathy, where this is a plate where you get the virus product produced. Next, click, please. And you would need to treat one patient, 2,000 plates, to give intramuscular delivery for the first clinical trials in Duchenne myopathy. So next slide. This is today a child treated IV for Duchenne myopathy. Next slide. And next slide, please. And this is, and you can click another time. And this is a child. Click, please. And this is a child before and after treatment. So just to say that, again, it works. It works, one administration IV. But how do we do that to treat hundreds of patients with Duchenne myopathy? So next slide. This is the kind of dose we need. How do we do to produce this dose? Production needs will be over 10 to the 20. This is not feasible easily. So we need to develop new strategies to be able to do so. Next slide. And this is what is currently done by many companies and many labs to improve this and to be able then to deliver to the patients. Next slide. Next slide. Okay, so this is the kind of technology, but it's, of course, strongly improved, so we can really improve the yields and be able to cover the needs that we will have for the patients. Next slide. So just to comment on that, you may not see this because it's very small, but it said that you should store that at -65. How do you do to treat hundreds of patients around the world in many countries if you need to store your product at -65? So again, this is where we need innovation. How can we move these products to some product that can be stored more easily at room temperature and be used all around the world. So this is another question. And this is addressed today, and this is going to change. Next slide. Okay, then next things, brain and complex disease. Next slide. So this is brain. This is what we do when we treat patients for this lysosomal disease, or tomorrow for Huntington disease. And this is Michel Zerha, the neurosurgeon who is in my team and who is doing that. And interestingly, I have the great opportunity to work with the only neurosurgeon I know who is willing to do everything but not neurosurgery. So what we are trying to do now is moving from neurosurgery to something that could replace neurosurgery, not to have to go into the brain and put a needle in the brain of patients. And this is an example of what we're doing by using ultrasounds to try to combine with IV delivery, open the blood-brain barrier, and be very safe to the patients. Next slide. So now a word on an important point, going from rare disease to complex disease and to treat a large number of patients. Next slide. So it can be achieved by identifying pathological mechanism of disease. And this is important because if you identify a common mechanism that can treat several diseases, you can treat very rare disease. So you don't have to develop one product for few patients, but you can have one product for many diseases. And this, of course, is important for the future. Next slide. And of course, this is also true for complex disease, not only for rare disease. Next slide. So I will just show an example of that in what we are doing, which is targeting a pathway, which is the cholesterol pathway into the brain. I don't go into details. Please click, because there's a lot of click and click. So showing the relevance of that. So this pathway is affected in many neurological diseases. So we've been in this pathway to try to use it to treat diseases. And this is what we've done. Next slide. And based on that strategy, we've been able, for a very severe disease of the brain called Huntington disease, it's chorea. So you may have heard of this disease called chorea. It's a very severe genetic disease, to demonstrate in vivo, in mouse, that this is efficient and this can cure the disease. And based on the results from the preclinical work we've done, next slide, we are starting a clinical trial next year to deliver the therapeutic gene that is restoring the metabolic defect in this disease. But interestingly, next slide, what we have showed, click please, click again, is that the same mechanism of action is effective not only in this Huntington disease, but on others, on Alzheimer's disease and on other, which is not genetic, and on other genetic disease called spinocerebellar ataxia. Next slide. And next, go fast because we don't have time. It took, back, just the one back. Okay, so, and based on the preclinical work that we've done and the understanding of the pathology that we've made, we've been able to address all the disease you've seen here. Huntington, spinocerebellar ataxia, Charcot disease, so amyotrophic lateral sclerosis, and Alzheimer's disease. And all these share common mechanism. So one treatment can treat several diseases, not only rare disease and also very rare disease like spinocerebellar ataxia, but common disease like Alzheimer's or amyotrophic lateral sclerosis. Next slide. So pass, because I see you're looking at your watch, so I'm, okay, so pass that, this is Alzheimer's disease, click, okay, this is spinocerebellar ataxia, and click, this is, the only thing important is here, it's spinocerebellar ataxia is a group of very rare disease, so you cannot develop one gene therapy for each of these very rare diseases, but this strategy is working for all, so one product for many diseases. This is the future. Next slide. Okay, so this is the end. So you've seen the progress that we've made, and you've seen the number of products that are in development or are already on the market. So it's really classical medicine, gene therapy has become classical medicine. Next slide, and I think it's the last one. Okay, click. And just to say that Again, it's become classical medicine. It's going to frequent disease, so it will treat a lot of patients, and because it will treat a lot of patients, the cost will decrease. And also, because it will treat a lot of patients with complex disease, it will have to go back to rare disease and be able to bring this treatment also by decreasing cost and improving technology to rare and to also very rare disease. And I think this is the key for the next years. And I think there's just one more, and I think I'm done. No, that's fine. That's the people. That's all the people that work on it. I'm sorry for the problem with the slides, but
No problem.
okay. Is there no question now? Okay.
Thank you. I think a good presentation to complete what was said by Frédéric before, and to introduce the other presentation, you know, and very, very positive messages and many questions for the roundtable at the end, you know, many questions. So now the third speaker is Marina Cavallana from the Necker Institute, Imagine Institute. Thank you to be there.
So thank you very much for this kind invitation, and thank you especially to Alain Herrera to have organized these innovation days that are very fruitful for all the participants. So I would like to stress, first of all, one important thing. Cell and gene therapy are not a single discipline, but are integrated medicine. Cell and gene therapy are science. And if you don't integrate all the concepts from physiopathology of the disease up to production, you can't make a very good cell and gene therapy. So from this statement, I thought recently to found a startup where we try to integrate all the scientific, medical, and economical problems raised by cell and gene therapy, and this startup is called Smart Immune. So they told me that it works, but it doesn't work. So the rationale to create this startup raised from the previous one, please, by the knowledge of the need of millions of the people around the world in terms of definitive cure. Hundreds of thousands of patients around the world need allogenic bone marrow transplantation. Unfortunately, very few of them have a HLA gene-compatible donor. So in most cases, as a physician, we are obliged to choose the best one we have to try to fix the problem and cure the patient. The more closest you have is one family members, unfortunately partially compatible with the recipient. In this case, as illustrated in this slide, the patient go to the hospital, receive a very huge conditioning regimen to ablate the stem cells compartment, but even eventually the cancer cells. And then he is grafted back with a donor stem cells. Unfortunately, it begins at this start point. It's fight against opportunistic infection, fight against immunological complication of allogenic bone marrow transplantation as acute graft versus host disease or rejection. And the patient can stay hospitalized 1 year before the only very useful hematopoietic cells are generated again. Which are these cells? These cells are able to fight against a very life-threatening complication are T-cell lymphocyte, the adaptive compartment of immunity. Next slide. So we worked in our institute for years to try to understand the track from the bone marrow up to the thymus and again in the bloodstream of T cells. And years after years, we have set up a very simple way to reconstitute in vitro the thymus compartment. We are able now, starting from stem cells, undifferentiated, and coming for whatever the source you wish to have, a culture system lasting 7 weeks, able to mimic the first step of intrathymic maturation. We have demonstrated, if I can have the next slide, that if we put the cells in this system, we are able to obtain what we call pro-T cells. These pro-T cell CD7 positive are able to migrate it into the thymus of non-Hodgkin gamma-C immunodeficient mice and give rise in a very shorter time period a complete adaptive system. We have demonstrated this work hypothesis in an immunodeficient mouse model with all the scientific tools we are at our, they are available in the field. So RNA-seq to demonstrate that these pro-T cells have closed all the transcriptional program necessary to produce other hematopoietic lineages. And of course, these cells are not able to give rise to any immunological complication. Immunology is really an important component of cell and gene therapy. Without a very solid knowledge of immunology, you can't successfully conducting any cell or gene therapy trials. Next slide, please. So these pro-T cells are able to produce an adaptive system, a polyclonal T cells in a very fast way. These T cells that have finished their maturation into the thymus are unable to produce any of the negative effects that face the patient after an HLA partially incompatible bone marrow transplantation. So, for example, these cells educated into the thymus are tolerant towards the MHC complex of the recipient as well as of the donors, so no acute graft versus host disease are expected. These T cells are polyclonal, so able to fight against all the opportunistic infection that the recipient develops in the first 3 months after transplantation. They stimulate the engraftment of the stem cells, and they have, as you very well know, a very long lifetime. So they protect the individual all the time required for the generation of T cells from the CD34 stem cells engrafted. You probably know that the time necessary from stem cells to go to be primed in the bone marrow, then to go to the thymus again, sort out as T cells is a very, very long period. These take up to 12, 18 months after transplantation, and in this period we lose 30% of our patients. Next slide, please. So our process that we have patented and reinforced with several one licensed today to our company consists to shorten it in a significant way this fragility period lasting so long, and to bring to the patient the possibility to rebuild their immunological system and go home in after the first few months after the transplantation. So this is the goal of our procedure and also of our startup. Next slide, please. The other bottleneck is scale up and scale out. So it is impossible that you can call you Novartis, Sanofi, each big pharma, you know, but if you are not able to bring this success operation at the bedside, you can't be successful, because you can't build a huge industrial center of production where you produce CAR T cells, gene therapy, because it is impossible to fulfill all the traceability needs you have to make this treatment sure. So our idea that we developed in a partnership with Univercells is to bring the innovation at the bedside. We can do it with a sort of a very small machine that is not a requirement, very sophisticated GMP laboratories, but that everyone can easily use. We deliver the machine with a very useful soft program. We are able to educate the personnel, the staff in the hospital to do him themselves, and they can deliver at which time, at which moment the physician wishes for his patient. This machine can be also solve the problem of how we can take innovation all around the world, not only in North America or Europe, the very rich countries, but also in countries with low or poor income, as Brazil or Asian countries. The next slide, please. So this is the description of our ongoing clinical trials. One clinical trial for SCID patient. It is impossible to think that we can produce a gene therapy for any single disease. And I appreciate a lot the slide of Nathalie Cartier that just the speaker before me, because we have to find strategies to cover more largest the population that need innovation. So the example she provided with this comprehension of physiopathology that allow to treat a different gene defect is the good one. So this is the reason also we can't build up SCID gene therapy for each 20 different molecular genetic defect that characterize this disease, but we can solve all the problem in once if we are able to solve the problem of haploidentical bone marrow transplantation. We have treated the first patient. Of course, I am not allowed to provide you with our result. And the second trial is ongoing in our hospital for acute myeloblastic leukemia with a W cord blood, one manipulated and one unmanipulated. The trial is just approved by our regulatory agency, and we start very soon. Two other trials for acute lymphoblastic leukemia for children. The patient are being started very soon, before the end of this year in New York at the Memorial Sloan Kettering hospitals. The next slide, I would like that we have fixed all the characteristics of the drug product we are able to deliver to the patient in terms of phenotype and molecular characterization, the cell quantity, the ability to manufacture and scale up very quickly for all the patient need, and the source of the stem cells. This project is a very comprehensive one, able to answer a number of questions also raised by the CAR T-cell technology, because the scale-up and the production of a million of CAR T-cells is a bottleneck for a number of industrial partners. We are able to obtain pro-CAR T-cells, able to go to the thymus, to proliferate, to have a very long lifetime that can solve the problem of exhaustion and declining of the CAR T-cells in the peripheral blood. So science, again, is the basis of all the innovation. The next slide, please. So the science has permitted us to really set up a thymic condition that they mimic the physiopathology with an enormous plus value consisting in our patent and knowledge. Clinically applicable to a number of diseases that can be genetic or acquired, with the idea to overcome all the bottleneck we face as a physician to the allogenic bone marrow transplantation. So this is our pipeline, and I would like to stress that we have obtained recently from EMEA the ODD for the first indication of this pro-T-cell drug product. And I think this has been obtained in the time record when the science is well-founded. Please, the next one. Probably. This is the last one. So I am ready to answer later on all the questions, and I hope to have not...
It's very pleasant to see a scientist wishing to start a company or starting a company. When I started my first company 40 years ago, it was not very common, you know, and it was a long time ago. Thank you. So now we have, but we'll see that many scientists are starting companies now, which is very, very optimistic for the future. The next slide is for Sebastian Amigorena. I saw him. So he's speaking from his office. Can we have him? Sebastian Amigorena. We saw his face before.
Hello, I'm here. Hello? Can you hear me? Well,
What is happening?
hello? Oh, I hear the echo now. I am connected and my camera is on, so I don't know why you can't see me.
No, cannot see you.
So shall I start without a video?
But we need the slides also. Yes, we have. So perhaps you can start to speak, because to save time and to help them to resolve the technical problems.
Okay.
Start,
So
yes. Thank you.
I will tell you about a lab discovery that was made at Institut Curie in my team, which is related to epigenetic programming and reprogramming of CAR T cells. Can you give me the next slide, please? The one after that. So the chromatin has a dynamics of its own that is very important to implement gene expression programs in cells in general. The pathway I'm going to discuss today is the one that converts heterochromatin into euchromatin and vice versa. This equilibrium is extremely important to implement gene expression programs in different biological situations. The molecule, and I can have the next slide maybe, I will be discussing today is called SUVAR 39H1. I see that one arrow is missing here. So SUV39H1 is not involved in the conversion of heterochromatin into euchromatin, rather the other way up, but the arrow is missing. But it is involved, it is regulating this process and the dynamics of conversion of euchromatin into heterochromatin. And so it's a silencing molecule, it's a negative regulator of gene expression. And we started working on this in collaboration with Geneviève Almousny many years ago and trying to understand how this molecule is involved in the regulation of immune functions. And a couple of years ago, we made the following observation that is illustrated in the next slide, and which is related to T-cell memory. Can I have the next slide, please? Yes, thank you. So this is a schematic representation of T-cell differentiation. Naive cells are activated into activated T-cells which cycle. And during this proliferation, the cell has to choose between two fates. Either it becomes an effector, which are short-lived cells that kill targets, in the case of CD8 positive T cells, and then die themselves. So it's a short-lived effector. Or they differentiate into memory, that is the blue pathway in the bottom of the slide here. And these memory cells can redifferentiate. They are plastic, so they can go back to proliferation and effector, but they can especially and very strikingly survive for years and years. And so we showed that SUV39H1, this epigenetic regulator, is involved in the silencing of the stemness and memory properties of differentiating T cells. So you need this molecule for an effector cell to become short-lived and to disappear after it has killed the target cell. We published this a couple of years ago, and this was done in an infectious model, listeria, in mice. And actually, the observation that triggered the initiation of our biotech adventure, the startup that we founded, that I will describe in a few moments, was the one that is illustrated in the next slide, which came when we implanted tumors in mice that are knockout for this molecule. And what we saw is pretty striking. It's illustrated on the left panels that show tumor growth after treatment with anti-PD-1, with checkpoint inhibitor. And what you can see is that the anti-PD-1 in this particular model is not completely inhibiting tumor growth, but it is delaying tumor growth, and that is the blue curve on the upper right panel here. When you implant the same tumor in the SUV knockout mice, and that is the lower left panel in gray, you see that the tumor growth is delayed, not as well as with anti-PD-1, but it's an intermediate situation. But what is very striking is that when you treat the mice with anti-PD-1, now the mice respond much stronger, and actually over half of the mice have completely rejected their tumor, and that is illustrated on the right lower part of this panel, where in the orange you see the synergistic effect on tumor growth between the SUV knockout and the anti-PD-1. So it is based on this specific result. We analyzed the mechanism and we understood what was going on, but it's based on this single experiment that we decided to go to human experiments. The next slide sums up very schematically what we think is going on. Next slide, please. Yes, so basically what this work shows is that SUV39H1 is involved in the differentiation in the upper part of the panel, in the differentiation of T cells into effectors. That is what I showed you before in the Listeria model. The tumor model suggests that the same molecule is also involved in the lower part of this scheme, which illustrates what happens to T cells when they enter tumors. They become exhausted, they lose their functional capacities, and SUV39H1 is required for this loss of T cell functions and this exhaustion process. So when we do a knockout of SUV, we actually get earlier cells that are less differentiated, less committed. They cannot install this epigenetic barrier that makes them completely exhausted, and PD-1 reprograms them and brings them into the upper part of this scheme. So we went to look for money to try to implement this and put this into humans, and we found a fund that was willing to do that, and so they told us, okay, please show us that this also works in humans, which made a lot of sense. And so we set up to do that. The next slide shows what happens when you do this exact same experiment, so knocking out SUV39H1, this time in human CAR T cells. So the CAR T is a CD41BB, CD19 model, so it's a very experimental model that is used very largely. And you see in blue and red the difference between the mock knockout cells and cells where we knocked out SUV using CRISPR-Cas9, the red. So the result was very spectacular. It lasted for very long, and we could even show that in the long run, so day 73, this is the right part of this panel, you can see that the cells persist for much longer, and this is a 2-log difference in the number of cells that we found in mice after 70 days. We did this in different flavors, in different models, and it is based on this result that we are now trying to extend and to refinance the company to bring this to the clinics. The next slide shows the company that we had founded after. So Alain Maillard is the CEO, and we got Michel Sadelin interested in our project. He repeated, or he did the experiment with the SUV knockout. He was very excited about the result that he got in his particular model with a CD28 CAR T cell, and with this team, we are in the middle of refinancing this Memo Therapeutics, which is the name of the company. In the next slide, you can see an illustration. So what I just showed you is this epigenetic programming. The company has a series of other assets, including a series of tools that Michel Sadelin has developed in his lab, and he put the property for, and he licensed his patents, which includes a new structure of the CAR T cell that allows to target antigens that are expressed at very low levels in T cells. And the reason why this was very important is because another part of our company is this specific tumor targeting platform that we call e-antigens, that identifies a new family of tumor targets that are extremely specific. This is associated, and is the last thing I will tell you, to a very unique and internationally recognized cell production center. So Isabelle Rivière is also a founder of the company. And so our program is to develop the platform first with known targets, and then to come up with our own specific e-antigen, shared tumor antigens. I don't have time to explain you what that is. It's a very exciting family of tumor-specific antigens. So I don't think I have another slide. I thank you very much for your attention, and I really wanted to thank the organizers for putting all this together and for inviting me to this presentation. Thank you.
Thank you. Thank you, Sebastian. So now the last communication concerning gene therapy from Mohammad Moti. So Mohammad, where are the slides? Is Mohammad connected? Yes.
Yeah, good morning, everybody. First of all, thanking the organizers for inviting me. It's really a true honor. Unfortunately, because of these COVID issues, I wasn't able to be in person, but really congratulations for a wonderful event, which is really needed in our field because, as I have put in my title, and as you have heard earlier this morning, this is a true revolution in the field of oncology. Next slide, please. So just to, for the sake of introduction, summarize the way currently we view the paradigm shift in oncology and hematology. Actually, for 3 or 4 decades, we've been living with the conventional chemotherapy, just targeting the tumor cell itself and trying to destroy it. In the late 90s, we started seeing the antibodies, and we have to admit that one of the biggest advances in hematology is actually rituximab, the anti-CD20, because you can cure non-Hodgkin lymphoma with a few cycles of rituximab. And I think this is really, if you add also TK inhibitors, especially imatinib in chronic myeloid leukemia, which is very orphan disease, definitely, I mean, these 2 treatments have been a true breakthrough. However, progressively, we have seen the advent of immune modulation, immune targeting to target the host. This is about vaccination. This is about the immunomodulatory drugs like IMiDs, which are extremely successful in multiple myeloma. And last but not least, although they have been extremely powerful in solid tumors, but still even in hematology, we started using checkpoint inhibitors. And now I think the paradigm has moved into a sort of a next generation era of immune cellular therapy. And by the way, when it comes to cellular therapy in hematology, and I'll come back to the history, it's really a relatively old story because the first version, I would say, of the software has been the allogeneic stem cell transplant procedure. And we heard a bit about this earlier today, very elegantly summarized by Dr. Cavazana. Next slide. So I don't need to convince this audience, I think, that the use of the immune system is extremely powerful. It can be specific. It has some long-lasting memory, and it can become a sort of a universal approach to many cancers. Also, it looks like, and we'll talk about this later, that some hematologic malignancies are maybe slightly easier to target compared to other solid tumors. Next, please. Next slide. Okay, just for those in the room who are not always familiar with the developments in hematology, actually the story of manipulating the immune system and the donor lymphocyte has been really pioneered by the first era of allogeneic stem cell transplantation. It was in 1957 when Don Thomas, who received in 1990 the Nobel Prize for allogeneic stem cell transplantation, clearly wrote that you need to manipulate the lymphocytes in order to cure leukemia. Then progressively, we had some important developments, including the fact that we can simply reinfuse donor lymphocytes, so these are healthy lymphocytes from the stem cell donor to a patient and allow remission. And interestingly, this has been progressively combined with all other tools of immunotherapy. And we had in the 80s and 90s a big era of cytokines. They're less popular today, but they were thought, I think, during my training to be really cutting edge discoveries in the field of cancer. Next slide, please. CAR T cell, I think I will not spend a lot of time to explain this because you already heard about it. I wanted simply to insist, and I believe this is an important thing in the field, that although we're talking about cellular therapy, actually now it is also viewed as a true drug. And most importantly, and maybe this is the limitation, the barrier, it's a living drug. So theoretically, over the long term, this drug is supposed to persist. So with a single shot, you are supposed to introduce a sort of an immune surveillance that should last forever. However, the reality is not the same, because we know very well, and this is the reason why patients continue to relapse after CAR T cells. The reality is that persistence and expansion in vivo is really a limiting factor. Next slide, please. Next slide, please. Yeah. Another important aspect, and I am showing this slide for the purpose of this meeting, and simply to highlight to all stakeholders, but also to the investors, because obviously we need financial resources to develop and to drive these innovations, that the development of CAR therapy, which is viewed very fancy and very timely today, took actually more than 3 decades. So although we live in an era of, Twitter and everything should go quickly, and email, and you have to solve things, in a few characters and in a couple of minutes, actually we have to admit, and I don't believe things have changed, that it can take many, many years between the initial observation, the initial idea, and treating a patient or getting a label for a drug. So the counterpart of this is that we need commitment and people willing really to have some persistence over the long term. It can't be like, I'm trying for the next 6 months and see what's going to happen. So it's really an important message, and this is why meetings like this one are extremely helpful to put all stakeholders together to realize these requirements. Next slide, please. So this is just to highlight the huge and amazing clinical development. I didn't update this slide. This is from May 30, but even with the COVID era, actually the number of clinical trials continue to increase. Next slide. So how CAR T cell works, next slide. So for us in the clinic, and this has been also mentioned, I think, earlier today, logistically, it's a bit quite complicated to manage, because this is a personalized medicine, personalized drug. For the moment, we are using exclusively autologous CAR T cells, although we're performing clinical trials with allogeneic CAR T cells. So the patient would undergo apheresis to get the T cells isolated. They will go through the engineering process in the lab, so ex vivo. So they will be shipped to specific facilities. And you would imagine that during the COVID, for instance, that has been quite problematic, especially during the peak of the pandemic. They have to be modified. They come back in a bag and they are reinfused. So all of this logistical process is actually quite complicated. Also, maybe scientifically, it's not a big deal, but actually putting all this together from step A to the infusion can become a bit of a challenge. But also, because it takes time, it is a limiting factor, because while you are waiting for your CAR T cells to be prepared and engineered, actually these patients can relapse. Next slide, please. Next slide, please. Okay, so the structure of a chimeric antigen receptor is, I think, well known to everybody. What I wanted to highlight, next slide, please, is simply the endless number of variables to be considered for CAR therapy. These are just some of the criteria, and we heard a very nice talk from Sebastian a few minutes ago about the epigenetic modulation. And actually, when you put all of these parameters together, the way you design the CAR T cell, the choice of the target, the transgene being used, the cell target, the expansion conditions, the gene transfer, the clinical management, the conditioning therapy, eventually any adjuvant post-infusion therapy. When you put all of this together, you have an endless amount of combinations. And I believe we're still today at the first generation of these CAR T cells. And this morning I have seen already publications about bispecific CAR T cells. Next slide, please. So one key issue for this to design CAR T cell is to identify the specific tumor antigen. And that's really a very complicated part because it's not very easy to have very specific tumor antigen. This is why, for instance, CD19 has been extremely popular, because we're fortunate enough in hematology to have this widely expressed antigen where you can have an antibody. It showed efficacy in vitro and in vivo, and most importantly, the safety, the absence of cross reactions against normal tissues. And that's really a huge barrier. And this is why, for instance, it has been a success in lymphoid malignancies like lymphomas and acute lymphoblastic leukemia, which are CD19 positive, whereas in myeloid malignancies, acute myeloid leukemia, we are still at the infancy because of the lack of this target where you would avoid attacking normal tissues. Next slide, please. So next slide, please. Where do we stand? Commercially, we do have two products, Kymriah and Yescarta. They are approved in non-Hodgkin lymphoma. So non-Hodgkin lymphoma, this is 12,000 new cases every year in France. Hopefully, half of them are likely to be cured, just first-line therapy without the need of CAR T cells. But you would imagine that this is a growing market, unfortunately. But also, because these are diseases of the elderly, the number of cases will continue to increase. Acute lymphoblastic leukemia, next slide, please, is also an indication. And the next label, and the FDA has granted recently a breakthrough process to this. There are many CAR T cells now being developed in multiple myeloma, and most of them are targeting the BCMA antigen, the B cell maturation antigen. And I think the initiation of CAR T cells in myeloma will really be transformative, because actually all myeloma patients theoretically will need CAR T cells because they all relapse. So while you have 5,000 multiple myeloma patients in France per year, 30,000 in the US, well, actually 100% at some point will receive them. So that's going to be a huge logistic for us, for instance, in hematology department, because the number of these patients that will be eligible is going to be very important. And I'm not alluding this, maybe we can discuss this later on if you wish, but this is creating a lot of burden on the functioning of the hospital, on the availability of beds, which is not yet anticipated, unfortunately, including ICU beds. Next slide, please. So this is actually my conclusion slide, that I view it in a positive manner, but these CAR T cells are here to stay. But actually, these CAR T cells are really the first generation. And now we're talking, as I mentioned a couple of seconds ago, this morning, the Nature Medicine paper about bispecific CAR T cells. So all kind of modulation. CAR T cells, but also CAR NK cells. They were published last year in the New England Journal of Medicine, and we will be just starting soon a phase 1 trial in France with CAR NK cells. This is the product initially from MD Anderson and now being developed by Takeda. CAR gamma delta T cells, I'm aware they exist, but even it's probably expanding beyond the oncology field, because you may also engineer and produce CAR regulatory CAR T cells. So you would imagine the whole autoimmune field. So this is the short summary I wanted to provide today. I know these were really very basic issues, but I thought it allows to position the problem, and I'm more than happy to take questions later on. And I do thank you all for your attention and wish you good success with this wonderful event.